Hello students, welcome to the class of Botany. Today we will be dealing with again one of the very important topic, that is your cell cycle and cell division. Agreed, it's a bit struggle in order to memorize, in order to identify that what difference is, or how can we distinguish between mitosis, meiosis or amitosis. So today let's make this lecture in order to simplify it so that you will be able to memorize it. So let's start with the normal definition that we come across. When we talk about cell cycle, cell division, what so far we have learned that yes, cell cycle, cell division deals with your mitosis and meiosis. Mitosis being your equational division which happens in somatic cell, and meiosis being your reductional division, that means getting haploid number, and especially it occurs in gametogenesis, that is your germ cells in plants and animals. Agreed? So taking that knowledge ahead, let's dig into the more important things that we have to do today. So let me slide the board for you. If you can see here, we have started with the definition of cell cycle. So the cell cycle, simple definition, it's the sequence or the events by which a cell duplicates. I'll just do it like this. By which a cell duplicates its genome, synthesizes other constituents like proteins, and then eventually divides into two daughter cells. This is the term which defines your cell cycle. Now when we are talking about cell cycle, who was the first person who identified cell cycle? It was Howard and Pelk. It was Howard and Pelk in 1953 who actually worked and identified cell cycle. And yes, also cell cycle cannot happen like that. Cell cycle needs some help. So the general protein which is there in order to carry out the cell cycle phases like your mitosis, meiosis, we have your cyclin protein and one enzyme kinases which is cyclin dependent. Again, the two important things, one is your protein cyclin, and yes, the cyclin dependent enzyme which is your kinase. Okay, so now let me quickly rub it off so that I can tell you more precisely about cell cycle and cell division. As you can see, I have already said that we have discussed, or people know about it, cell cycle, cell division includes mitosis, meiosis. But yes, there is one more phase which it includes. So just let me do it like this. Let me make a bubble here. And let me make another bubble here. When I'm saying bubbles, I'm putting something here in it. Okay, let's see. Cell cycle or cell division generally has two phases. Interphase, and then there is a M phase. It has interphase and it has M phase. Now let's see, what does it have? Interphase has G1 phase, which is also known as gap phase. It has S phase, which is also known as synthesis phase, and then it has G2 phase. It has G2 phase. This is the major one which deals with your interphase. Interphase is that phase in which the cell is resting. That is why interphase is also known as resting phase. Interphase is also known as resting phase. So interphase is that phase in which the cell is resting, cell is preparing itself for cell division. Cell is preparing itself for cell division. So during the preparation, it will go through G1, S and G2 phase. When I'm talking about your G1 phase, as the name suggests, gap phase, of course, between two successive cell division when there is a small period of time, that can be concluded that we are talking about your G1 phase. Also, G1 phase is the phase in which the cell is growing. Cell growth occurs. Cell growth occurs. That means cell is preparing to undergo cell division. Cell is preparing to undergo cell division. Then we have synthesis phase. This phase is very important. This phase is very important. Why it's important? Because this is the phase during which DNA replication occurs. DNA replication occurs. Okay? Or you can say duplication of DNA. But the number of chromosomes remain same. But the number of chromosomes remain same. When I say number of chromosomes, I'll tell you what do I mean by this. Say for example in your G1 phase, say for example, I'll just quote the example here. Say for example, in your G1, I'm talking about DNA replication occurs, but chromosome number remains the same. What do I mean by this? Say for example, during G1 phase, number of chromosomes were 2n. Number of chromosomes were 2n. But even when it enters S phase, still it will be 2n. But duplication of DNA occurs. And this is very important question because this question has come up in your competitive examination, that during which phase of cell cycle, cell division, DNA replication initiates, or when does DNA replication happens. So it is your S phase, which is also known as synthesis phase. So now let's move ahead with your G2 phase. When I'm talking about your G2 phase, during your G2 phase, generally what happens? Of course, DNA replication is happening, DNA duplication is happening. It happens in nucleus, whereas centrioles duplicates in cytoplasm. Centrioles duplicate in cytoplasm. Okay? So now let's move ahead and fill up this one. I might need the boat, so I'm going to rub it off. Now let's talk about your M phase. When I'm talking about your M phase will generally, it can generally be categorized as simple, mitosis or meiosis. Mitosis or meiosis. Of course, mitosis is also known as equational division. Meiosis is also known as reductional division. And of course, this one occurs in somatic cells, and this one occurs in your germ cells, or you can say during gametogenesis. During gametogenesis. So in today's lecture, we will be dealing with just the introduction of your cell cycle and cell division, and then in the simultaneous lectures, we will be dealing with mitosis and meiosis separately. Let me give you, yes, when we talk about M phase, that is mitosis and meiosis, there is one more kind of meiosis, which is the primitive form, and it is very unique. Why is it unique? Because it's an exception. When we talk about exception in eukaryotes, because the eukaryotes are there, that means of course mitosis and meiosis will be there. So exception eukaryotes will be your yeast. So they might go under a very specialized cell division. I'm going to write it down here. Amitosis, which is primitive and an exception in eukaryote. The best example would be yeast. Now when we have started talking about the yeast, let me give you a quick important note on this yeast. Yeast is one eukaryotic organism which generally takes 90 minutes to complete its cell cycle. 90 minutes to complete its cell cycle. Whereas when we talk about your humans, so generally the cell cycle occurs within 24 hours. Within 24 hours. Okay? So now let's do further of this. Let's talk about your mitosis. So I'm going to write down few points of mitosis here. Let me tell you, let me write it down here so that it becomes easy. Let's do mitosis. I'm just giving you a general information. In the next classes, we will be dealing with mitosis in detail. So mitosis in animal cells and mitosis in plant cells. Let's know the name of the discoverer. In your animal cell, it was W. Fleming. It was W. Fleming. Whereas in your plant cell, it was Strasburger. You want to pronounce it burger? Let it be. Strasburger. So these two people, they identified mitosis simultaneously in animal cell as well as in plant cell. Okay. Now let's talk about mitosis deals with what? Mitosis, simple general structure, if I would say. This is my cell cycle. Okay, so when I say cell cycle, yes, the cell cycle is divided into certain divisions. This would be your G1 phase. Then of course you will have S phase. Then of course you will have G2 phase. And then you will have a period which is an M phase. That would be prophase, metaphase, anaphase, telophase, and then finally cytokinesis would be there. Let me draw this G1 phase a bit better here. So these are the major phases which actually take place during your cell cycle and cell division. Interphase, when we talk about the interphase, so that means from here to here, what I'm looking into? I'm looking into interphase, which generally happens or takes place, generally happens or takes approximately 95% of the cell cycle. So 95% of the time, the cell is only doing interphase, that is your resting phase. Now let me write down the names here. So this is, now I'm talking about the M phase. So let's start naming the M phase type. First one is your metaphase. Oh, sorry, not metaphase. prophase, metaphase, anaphase, telophase, and then finally you will have cytokinesis. So when we are talking about your cytokinesis, karyokinesis means nucleus division, karyokinesis. Once the mitosis or meiosis finishes off, they have to immediately undergo cytokinesis. Cytokinesis means now they will be dividing into two separate cells or dividing into the haploid or the diploid cells. So cytokinesis, cytoplasm division, nucleus division is your karyokinesis. There is a small structure here. That stage is known as G0. G0 is also known as quiescent stage. Now, what is your G0 stage? Okay. Now, when I'm talking about your G0 stage, a G0 stage is a quiescent stage. Generally, there are some cells in human body or in a eukaryotic cell which do not undergo cell division. Like say for example your heart muscles, okay? They will not undergo any cell division. Generally what happens is that they are active throughout the lifespan, but they will not proliferate. Generally it happens during the injury or any tissue damage. So what happens when the cell is in G1, it will not enter the S phase. It will immediately go to the G0 stage. And G0 stage means it remains active but it will not proliferate. Will remain active but will not proliferate. So from G1 to S1, instead of going from G1 to S1, it will immediately go to G0. So this is what is your cell cycle. So now let's come back here. So let me tell you the stages. We have already done the stages. Let me write it down one more time for you. Prophase, metaphase, anaphase, and then finally telophase. This is what is your mitosis. Let me rub it off and write down here meiosis. Now let's see what happens in meiosis. So when we are talking about meiosis, meiosis will be further divided into meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. So of course for meiosis 1 it will be prophase 1, metaphase 2, anaphase 3, telophase, prophase 1, metaphase 1, anaphase 1, telophase 1. What we need to take care of is that this prophase 1 is bit extensive. It's bit extensive. That means it is further divided into, let me keep on writing for you. Prophase 1 further divides into leptotene, zygotene, further stages, pachytene, diplotene, and then finally kinesis. Lazipadidae. Lazipadidae. they die. Okay, so this is the one. When we talk about meiosis, which is your reductional division, so meiosis 1 is further divided into your prophase 1, metaphase 1, anaphase 1 and telophase 1. Simultaneously, meiosis 2 will also have the same thing, but they will not have this. Only in meiosis 1, prophase 1, you will have certain divisions. Okay, when we are talking about your meiosis 2, it becomes like this. And it was Moore who observed, who gave the term meiosis, So students, this was the general information that we started with your cell cycle and cell division. So in the upcoming lectures, we will be dealing with mitosis, meiosis and amitosis in detail. Hopefully this general information is useful for you, and thank you so much guys.